Okay, so I'm gonna start by teaching just basics of using Python building function when we're working with biological data. So here I have my DNA sequence. I'm sure we all have seen the DNA sequence. I'm just hoping that we all know what the DNA sequence is. So I'm going to specify my DNA sequence, the variable here. And then the first thing that I'm going to do is try to print the DNA sequence. And then one of the things that you can do with building a function, Python building function when you're working with DNA sequences, you can be able to check the length, which is this line here. And you can also be able to count the sequences. You can be able to count each nucleotide that you find in your sequence, and also you can be able to find the positioning of whatever position that you want to print out. So you can be able to print that out. So those are just the basics things that you can do when we're working with DNA sequences in Python. Of course, this is great, but in real life applications, you're gonna need more than just this functionality, which is why my talk today is to introduce you to a Python package that's called BioPython. So it's a very easy to use Python package that we usually use for computational biology. So obviously the first thing that you need to do when you are about to work with a certain package is to install it. So it's very easy to install BioPython. You just you just run pip install BioPython on your terminal to install it. I already have it installed, so I'm not going to run this line. I've also included the official BioPython website where you can go to find out more about BioPython and also the documentation. They usually have everything that you need in the documentation, which is one thing that you should, if you start somewhere, just consult the documentation. It should give you all the answers that you need. Can I zoom the screen? Okay. Is that fine? Okay, I think this is the largest I can get. So the first thing that you need to do is obviously install the package, which is this line over here. So you can just copy and paste that line to your console, and then it should install the package for you. So while we're busy installing the package, I want to recap on a certain concept that we usually use in molecular biology. So I'm just going to change and share. Sorry, I just need to open my slides. So in molecular biology, we have what we call the central dogma of molecular biology. So it's basically how genetic information flows from one direction to another. So that's it. That's from DNA to RNA to protein sequencing. So it has three main steps, which is the replication, the transcription, as well as the translation. So the DNA is made of nucleotides, which is the adenine, the thymine, the cytosine, as well as the guanine. So this is what makes the DNA strand. So the adenine pairs with the thymine, and also the cytosine pairs with the guanine. So the whole reason why I'm explaining this concept is because I want to move to using to show you how you can use BioPython to move from this strand, which is the coding strand, all the way to the protein sequence. So how we do that is, I'm gonna have to go back to my slides now. I'm gonna have to go back to Python now. So basically, BioPython offers you modules and classes that you can use to work with biological data, such as DNA, as well as RNA sequence, like I've mentioned, and also protein and protein structures. It also like facilitates various functions in bioinformatics, where you can be able to use it to do sequence analysis, protein structure analysis, as well as to access and also interact with online databases such as the PubMed, as well as the NCBI. So you can work and manipulate data from various files, formats also such as the FASTA. That's the most common one. If you use bioinformatics, I'm sure you've came across a FASTA file. So you can also use it to access, directly interact with the online databases, as I've mentioned. It also allows you to work with protein data banks files, which is protein sequence analysis, basically. And you can also do phylogenetics and also population studies. But today, we're just going to focus on working with it, working with sequences, as well as accessing online databases. So obviously, after the installation, the first thing that you need to do when you're working with the package is to install whatever tool that you're going to be using that comes with the package. So BioPython comes with a sequence of modules. One of them is called BioSec, which is the one that we're going to be using to in dealing with these sequences. So the first thing I'm going to do is import the sec the sec class, which is the one that you use when you're dealing with sequences. And then the next thing that I'm going to do is have I'm just going to create a random DNA sequence. So I'm going to call that coding DNA. Since we don't have enough time, I think we should just copy and paste. So the package that you install, the package that you import, the first thing that you need to do is to install BioPython, and you install that by using pip install BioPython. And then the next thing that you need to do is import the sec tool that we're going to be using from the biosec module. So, and then I'm going to tell you to print my coding DNA. And then the whole point of me showing you the central dogma of molecular biology was just to show you how each nucleotide pairs with the next nucleotide, and also the different nucleoside that makes up DNA. So let's say now you have a coding sequence, a coding DNA sequence. You have the sequence, and then you want to find the complementary DNA sequence of that coding sequence. The first thing that the function that you're going to use is just called dot complement. It comes with the sec tool. So I'm going to do that now here. And then as you can tell, A pairs with T, T pairs with A, C pairs with G, and so on. Another thing that you can do is check for the template DNA sequence. And with that, we use a function called reverse complement, which is this one over here. And then you can also by just having the coding strand sequence, you can be able to also figure out figure out the messenger RNA that's going to be transcribed. So the function that you use for that is called dot transcribe, which is this one over here. So as you can tell, the sequence of the coding DNA is exactly the same as the sequence of the messenger RNA. The difference is that messenger RNA comes with uracil, and the the coding DNA has what we call thymine, a nucleotide called thymine. So you can also use it to find the protein sequence. It's basically going to give you the amino acids, which are the building blocks of the protein. So we find that by using a function called dot translate. So another thing that I wanted, sorry. So another thing that I wanted to show you is if you want to simulate mutation, let's say you want to change a certain nucleotides on your sequence. So because, sorry. So it's just like normal Python string, the seq object, which is the one that we are using here, is a read only. So apart from wanting the seq object to act like a string, it's default like this because we don't want to be changing the sequences unless you have to. And obviously there are some situations like when you wanna immutate mutation, then maybe you wanna change a certain a certain nucleotide on your sequence, like you want to substitute a certain nucleotide on your sequence, or you want to delete a certain nucleotide on your sequence, then we use what we call a mutable seq. So it's also a class that comes with BioSeq, but it's used when you want to change your sequence, because if you were to do it in a sense of just changing the position, substituting the position, then it's going to give you an error telling you that seq object don't work with, it doesn't work like that. So that's the whole function of the mutable seq class. So another cool thing that you can do when you are working with bio module, bio Python, you can actually be able to read your file formats, such as the FASTA, as well as the gene bank. But I'm just going to make an example with the FASTA file today. Okay. So someone is asking me to explain the mutable seq again. So just like strings, you know that you can just edit it. So the sec class also works the same way. So say now you want to imitate mutation, like you want to change a certain. Sorry, let me just not read slip because I'm getting this step. Anyways, let's say you want to change a subsequence in your sequence in your sequence. Let's say you want to change CTG into maybe ATG or ACG. What you can do then is use the index of wherever your subsequence is, and then sorry, if you use the index, because the normal way that you can change is just to use the index. But since sec objects only uses works the same way as string, you can't just change it like that. So the biosec has a mutable sec function that you can actually use for such things. So they call it mutable sec because they know that most of the times if you want to change your sequence, it's when maybe you want to imitate a mutation, for an example, or you want to change your whole sequence. That's why they call it mutable, just so you can remember easily. So that's the whole point of it. So another cool thing that comes with BioPython is being able to read different file formats that we usually work with in bioinformatics. One of those file formats is called FASTA. So a FASTA file So a FASTA file format is just a text-based format for representing either nucleotide sequences or peptide sequences in which the base pairs or amino acids are represented using the single letter code, which is ATGC. So a sequence file usually starts with a single line description where you find the header, and then that is followed by the sequence lines, which usually includes your DNA sequence, for an example. So I want us to download a FASTA file from the internet. So I know most of you probably don't have WeGet, so you can just use this line to install it. And then after installing, you can import it. Before you work with any Python package, you need to import it first. So this is what I'm doing here. So now we're no longer going to be using the seq class, but we're going to be using the sec.io, which is just input output, and that's because we're working with files. So from bio, you're going to import the sec.io. So this is the URL. I'm just going to copy it. I'm going to paste it on the chat. If you want the file, you can just So the SecIO input output contains a function called parse, which is PARSE, which usually takes your file path, which is this one here. So what I did here was I just specified my file, where my file is. And then when you're working with BioPython, we're gonna use this function, which is called parse. And then we're gonna input our FASTA file. It can be a path, or you can specify it as a variable there and then just call it here. And then because as I mentioned that it works with BioPython works with different formats of files, you need then to tell it what the format of the file is, which is what I'm doing here. So I wrote a for loop because if you ever seen a FASTA file, it usually contains more than one sequence of an organism. So here, the reason why I'm using a for loop to work with this, it's because I wanted to print everything that I need, which is the ID, the description, the sequence, and the length of each and every sequence that is contained within the FASTA file. And also, it's just easier to work with for loops. Makes your life so much easier. So that's why I opt to just use it here. So the first thing I'm going to do is specify my URL for my file. And then the next thing is I'm gonna specify my file, my FASTA file. Sorry. And then the next thing I'm gonna do, I'm gonna tell BioPython to print out the ID of each sequence on my file, the description, the sequence, the length, and then this line I'm just using to separate the records so that it can be easier to work with. Sorry. So what we're finding here is that my FASTA file, the one that I downloaded here, the one that I downloaded here, what I'm finding is that it has 11 sequences, different sequences for different organisms. So how they're specified, they're just specified as a seq1, seq2, seq3. But obviously, if you're working with sorry. Sorry, I have flu, so excuse me a bit. So if you're working with different sequences, okay, it's going to describe it for you. Like for an example, the sequence belongs to an organism named that, and the sequence belongs to an organism named that. But obviously, from wherever that you downloaded, the database that you downloaded your file, if you go to that database, then it's going to give you more information on what exactly the sequence represents. This is just what's usually on your FASTA file. So you can also use it to work with the gene bank file. Couldn't find one. I've never worked with them, but it's basically the same concept. You specify the file path here, and then you tell it that the format is the gene bank, and then it prints out for you what you ask it to print out for you. So obviously, sometimes you would wanna create a file, a FASTA file when you have like different sequences that you wanna store in a certain file. then you can use this method to create your own FASTA file. So this is defined in a module called BioSec record. It's also one of the modules that comes with BioPython. And then it uses sec record to it uses the sec record tool. Sorry. So same thing here, you're gonna define your sequence, and then you're gonna say the record. So you basically input whatever that is there from the side, because the side or your FASTA file is usually made of an ID, a description, a sequence. So obviously, if you're creating your own FASTA file, you have to have those things also. Yeah, and then you use sec input output to write it. Then you're gonna put your path that you want your FASTA file to be in, and then you have to also specify that it uses FASTA format. So as I said, if you want to be working with a certain module, a certain tool in Python, you have to install it first, obviously. And then after installing, you'll still need to import it in order for it to be able to work on your script. That's just how Python works. So here's the fun part. So one of the very common things that you're gonna need to do in bioinformatics is to extract information from the biological databases online. It can be quite tedious to access these databases manually, especially if you have a lot of repetitive work to do. So BioPython attempts to save you the time and energy by making online databases available from Python scripts. So it has different codes that you can use to extract different information from online databases. So one of the main databases that can be accessed is the PubMed. So I'm not sure if anyone has ever tried to access PubMed or NCBI. So if you've ever worked with accessing a certain database from online using, accessing NCBI database, it always asks for your email. So which is what the interest.email here. So you just specify your email, and then depending on what you want. So depending on what you want, like for an example, let's say you're working with sorry. Sorry, let's say you want to access PubMed. Let's say you have a certain topic that you want to research on, or a term rather, then you can just define the search term here. For me, I just defined the search term as a BioPython. Sorry. So the next thing is to give it a handle. So think of a handle as opening ports for yourself in the server. then you can use the connection to read or query the website or the database for what you need. And then here, this function here is just going to give you all the available databases that you can find. And then obviously the next thing will then to close the connection because you're done with whatever issue you're doing. So I'm just going to run this. Oh, and another thing I forgot to mention is that accessing databases uses a tool called entries. BioPython uses a tool called entries, which is why you have to import it from here. And then you're gonna have to insert your email address. This is just for record keeping. So I know that I shared the script with my email, so you can just use your own email. And then the thing I want to search for is BioPython. And then I'm opening my ports here so that I can be able to access the database. This part here is the one that you use to ask for how many databases, I mean to return the list of databases that are available. And then you close it. So you're just gonna you store the list of databases as rec. And then you're just gonna close the connection, and then you want it to print out. So I'm sure you've seen some of these databases. So we have PubMed, Contain, Nucleotide, Structure. So now you know that there is a database called PubMed within the NCBI. So the next thing that you would want to do is go straight to let's say you want to search certain articles that are related to a specific query. For an example, you want to search for all the articles that mention COVID-19 in South Africa. Then you can just use this. So I'm gonna store this in records. Now that I've got what I wanted, I'm just gonna close the connection. Then I'm gonna print out the PubMed IDs of the articles that mention COVID-19 in South Africa. So those are all the PubMed IDs of the right. No, I didn't actually register with this email. It worked either way. So, So you have the list of the PubMed IDs, and then now we're gonna use a function called fetch to fetch whatever data that we want from the PubMed. So again, you're gonna tell BioPython that this is your PubMed. Let's say you're interested in that article, then you're gonna tell BioPython that you wanna see what's in this article. Then you're gonna use the function ifetch to specify your to specify the database that you want, and then the ID of that article, and then the format that it should come back as, sorry. So in those last steps, you're just telling it where it should get the title, the abstract. Let's say you want to print out the title and the abstract. So you're going to specify it here. Sorry, let me just check. I'm reading. Okay, so got an error here saying that the PubMed article. Oh, so it seems like the the article number that I chose, it doesn't have it doesn't have a PubMed article as one of the records, which is why I got this error here. So I'm just gonna skip over that. Yeah, I think I missed one of the um one of the variables. Let me just check. Just gonna rerun the whole thing. Sorry. So So this is like the abstract of this title that I chose, which is the title, the article that I chose, which is an article with this PubMed ID. So it just shows me the abstract as well as the title. Of course, PubMed is not the only PubMed is not the only database that you would want to access, especially if you're a bioinformatician. One of the databases that you definitely want to access or inquire is called nucleotide, which is basically what I've been showing you the whole time. So it works exactly the same. The only thing that you need to change is the database name, as well as your search term, depending on whatever that you're interested in that moment. So yeah. So that's basically the end of the things that I wanted to show you when you're working with BioPython. So yeah, the speaker can take over.
Hi, Nomlie. There are a couple of questions on Slack. I'm not sure if you saw them.
Okay, yeah, we can go through them. Sorry, I shouldn't have done this today. Flu is really killing me.
Sorry. Can you see the questions, or do you want me to read them to you?
I didn't see that I was muted, sorry.
Okay, no problem.
Okay, so you can read through the questions, yeah?
Okay, I'm also still trying to open my Slack window here. But then there was one about how do we filter the records. Okay, so since the databases contain a lot of info, how do we then filter if you say you want only faster files instead of all records? I'm not sure what prudent means about faster files. Nomlie, are you still here? Oh, okay. So Nomlie is just having some technical issues. She'll respond to the questions on Slack, but we have a break in 3 minutes. Okay, thank you. We have a break in 3 minutes, so she'll answer some of the questions in the chats, and then we can take our break so long, and then we resume at 15:15, so 3:15. So you can continue putting your questions on the chat on Slack, and then we will take a break for now and resume at 15:15. Nomlie, if you can hear me, please answer the questions on Slack in the chat channel. And then I'll pause the recording for now.
I'm so sorry about that. My PC switched off.
Yes, that's okay. You can either respond verbally or you can respond on Slack because we are on a break now. Or you can respond after. Is that fine? Nomlie, can you hear me?
Sorry. Yeah, after the break, we can like do the questions.
Okay.
Thank you.
You can go ahead and answer the questions on Slack. You can see them, right?
Yeah, I can see them. Yeah.
Okay, perfect.
I'm just gonna go for them now.
Yeah, you can go ahead.
So the first question, if we use string operation to split DNA strands into single strand, is it more than okay? To split to single strand. So no, the string operations that we use in Python are not there to use to split like the strings, but rather the sequences. So we're not really splitting the strings. We using one string to split out the sequences in that string. I hope that, and it applies for RNAs. For RNA sequences, it's the same thing that we're doing. We're not splitting the strands, but we are just analyzing the sequences that you find in one of the strings, like in DNA, or RNA only has one strand. I hope that answers the question. And then the next question is from Prudence. Okay. technique do I use? Sorry. So obviously it depends on the kind of data that you're working with. So this is very broad. I can't really answer unless if you're talking about a certain data type. But yeah, if you're working with biological data, the first thing that you need to do to identify the patterns, you need to do the pre-processing step. Obviously, that includes quality control and also removing lowly expressed genes. For example, if you're working with genes, sorry. And then if you're doing differential gene expression, then you can do what we call variant calling. And also, if you're working with pathways, then doing clustering would be one of the things that you would do to identify the patterns. And also you can use machine learning for classification. I'm not really skilled up with machine learning, but it's one of the things that you can use. And then the next question is working with high throughput sequencing. So obviously the main challenge is large volume of data, but we have CHPC for that. And also there's also Derisa if you want to store your data for long term. I've never really worked with the data large enough that I couldn't work with it, work on it on the cluster. So yeah. And then for visualizing biological data, it depends on what you're working with. But a heat map is one of the things that you can use. Also use it in my master's when I was working with differential express genes. Another thing that you can use is volcanic plots. I remember I had a diagram where I had to represent differentially expressed genes and genes that are not sorry that are not differentially expressed. So I used a volcanic plot for that and it worked out fine. Yeah, you can also, if you're working with pathways, then you can use pathway maps. So yeah, I hope that answers the question. The main important thing when you're working with biological data is the pre-processing step, because obviously, oh, and also the quality. It's very advisable to use databases that are widely used. And also, when it comes to tools, also, it's very advisable to use tools that are well documented, so that you can be able to get help if you need help, because obviously if it's used by many people, then many people can use you, when many people can help you for it. So yeah, I think I've answered all the questions.
Okay, thank you so much, Nomlie. To the participants,